All right, so today you guys are going to be looking at water notes. So in your binder, you guys should have gotten these last week when I was here. I did put a copy on Google Classroom so that you can go through and copy the notes into what I have there, meaning the supplemental pieces that you see here. I also like to tape them just so you guys can hear me talk about them. I usually will add a few more things. And that way, if you have questions on it, highlight the areas that you're still a little confused on. So that we can go through it when we come back. This material you guys will see on your Chapter 1 test. Once I come back and we do a few of our larger labs, we'll have that Chapter 1 test. So it's kind of a lot of material, even though a lot of it may be review material, it's still a lot of material. So when we're looking at our water notes, and why we do water, it's because it's so important for humans. It's important for the human body. About 75% of the earth is water, and it's fundamental for life. If you think about our bodies, the human body, about 75% of our total body weight is water, but it's not all the same throughout your body. So about 20% of it is found within your bones, but when we go through things like brain cells, about 85% is going to be found within brain cells. So it kind of changes, and all organisms are a little different. So for instance, jellyfish that you may see at the beach when you're walking along the sand are about 95% water. And it also changes as you guys grow and develop as well. The amount of water within the body will also change. So we do know that water is the universal solvent. We've heard that many times over and over and over again. It's an aqueous solution. What this means is that water can dissolve more substances than any other liquid. We just said life originated in water. Think about things within your body. Cytoplasm, that gooey stuff that makes up your cells, holds all your organelles in place. Your blood plasma, that your red blood cells and white blood cells and platelets are going to flow around into. Intracellular fluid between these particular cells. And what we know is that life actually originated in water about 3.5 billion years ago. We actually have microfossil evidence for this. So not only is it important for living things and it makes up our body, it allows for transport of nutrients vital for life in animals as well as plants. And if we think about water, you guys are going to be watching a little National Geographic video one of these days as well that talks about water. And if you think about why water is so important to our planet, if you think about just one drop of rainwater falling through the air, it actually dissolves atmospheric gases. So as it's falling to the ground, it's picking up those gases, it actually affects the quality of the lakes, land, and rivers. So as that water gets picked up by plants and as those plants utilize it, those atmospheric gases are now within them. So when we have pollution, we talk about pollution, we can follow that getting transferred along as well. When we look at the water structure itself, we know that it is H2O. What does that mean? It means that it's made up of one oxygen molecule and two hydrogen molecules. This is a polar covalent molecule. And I love this question because you guys are like, oh, we've done water. And I always go, yep, water is a polar covalent molecule. What does that mean? And it's still a little confusing to you guys. So break down that word, polar covalent. And what we have is that it is unequal sharing. So we have unequal sharing of those particular electrons that we have. So with that one oxygen molecule, we'll look at it a little bit more down the bottom, we know that we have 8 electrons. Oxygen's got 8 electrons, hydrogen's got 1. The first ring of oxygen is going to hold 2, the second ring is going to hold 6. It wants 8 in order to be stable. So what is it going to do? Hydrogen that's got the 1 electron wants 2 in the outer ring. So they are going to share electrons. So hydrogen is going to share an electron with oxygen. So now hydrogen feels stable. This hydrogen is going to share an electron with oxygen. It feels stable. Now oxygen is stable because it has 8 in its outer ring, which is that second ring there. So this is a stable structure now, stable molecule. It is covalently bonded. They are sharing electrons as opposed to an ionic bond where they're giving or taking electrons and creating ions. However, it doesn't look like this. So I'm actually going to put kind of an X through this. It doesn't really look like that. It is not a linear molecule when it's stable, because even though they're sharing these electrons, oxygen kind of hogs it a little bit. And what happens is that oxygen steals more of those electrons, those negative charges. And when it does that, it kind of pulls oxygen down and it will go through and kind of give you more of a V-like structure like what we have there. It is still covalently bonded. They are still sharing electrons, but now it is polar. It is unequal sharing, and that's what you guys see here. So the pink is going to be your oxygens, the yellows are going to be your hydrogen. Because of that, they're not creating ions, they're not creating charges, but they do have a slight charge to them, even though they're sharing them. So since oxygen is stealing more of those electrons, electrons are negatively charged, it's going to have a negative charge to it, slight negative charge. My hydrogens are going to have a slight positive charge. So once again, these charges are distributed unequally. Oxygen's negative, hydrogen has a positive charge. So now we have the kind of this dipole, meaning we don't create this linear structure. We got two opposite ends of this particular molecule. Because of this single water molecule that is covalently bonded together can hydrogen bond to another water molecule. So we're going to have two types of molecule or two types of bonding put together here when we start to create all these water molecules putting together. So hydrogen bonds just mean that we are going to see this weak little bond, and we will go from the, and I'll see this on the next page as well, the positive hydrogen is going to be attracted to the negative oxygen because opposites are going to attract. The thing about these hydrogen bonds is that they are super weak and they break very easily, but they are going to cause a ton of different properties that we love to see within water. And if you guys remember the water activity, or we'll do it when I get back, for those of you who haven't done it yet, you guys are going to be seeing these particular activities such as adhesion and cohesion and surface tension and capillary action and all that other stuff that probably is in the back of your head someplace, but you guys are going to look at it a little bit more. So as we go down just a little bit, we know that hydrogen bonds form between the hydrogen of one polar molecule and the oxygen of another polar molecule. So once again, these hydrogen bonds form between the hydrogen of one polar molecule, here's one polar molecule, and bonds to the oxygen of another polar molecule, a completely different water molecule. So we will be joining those water molecules together with a hydrogen bond, but each single water molecule is created through a covalent bond. They are sharing those electrons. So it gets a little tricky right there. So once again, single water molecule, covalent bond, that's what it is. We're sharing those electrons. Once again, oxygen's got 2 electrons in the first ring. It's got 6 in the outer ring because it's got 8 total electrons. It wants 8 in order to be stable. So in order to stabilize itself, it wants to either give and take electrons, which we don't want to do here too hard, or it's going to share electrons, which is a perfect opportunity because hydrogen's got 1 in the outer ring. It only wants 2. First ring can only hold 2. So if hydrogen shares 1 with oxygen, hey, hydrogen's stable. Let's do it on this side. Stable. Everybody's stable, giving us that H2O. Once again, it is not linear. Next page. And then I went from there showing you your covalent molecules to your hydrogen bonds. So a single water molecule covalently bonded, single water molecule covalently bonded, single water molecule covalently bonded, gives us slight positives and negative ends, and the opposites are going to attract. So those little pink bonds that you see there are my hydrogen molecules, or hydrogen bonds that you see there. So polar molecules that are attracted to water, think about it, they dissolve in water, they break down in water, such as sugars, are said to be hydrophilic, water-loving. Hydro meaning water, philic means they have affinity to. Non-polar molecules that repel water, they hate it, they don't want to be near it, it's not going to be breaking down. Water dreading, such as oil. So if you've ever cooked like a heated up big pot of water, maybe making pasta, and you put a little oil on top, that oil sticks together on top. It's not going to break down. That's said to be hydrophobic. So I just said that because of these hydrogen bonds, they're going to result in a ton of really cool properties. So these are ones that you guys should be a little familiar with, but here we go. So this hydrogen bonding, water molecules sticking together with these weak hydrogen bonds, create surface tension. And this means that water has a very high surface tension. The only liquid that's basically higher than water is going to be mercury. Mercury is actually a little bit higher in terms of that surface tension. And the surface tension causes things like cohesion and adhesion. Now for me, cohesion and adhesion, when I was in high school, oh yeah, I know them, but I really couldn't tell the difference between them. I knew they dealt with water, I knew they dealt with the surface tension and these bondings, but I didn't know the difference between them. So cohesion, if you guys have ever gone out to a lake or something, a pond, and you see these little bugs and they're walking across water, that's cohesion. These bugs are walking across those water molecules. They're not able to break those hydrogen bonds. They're light. You go ahead and you think, oh, let me try to walk across water. You've got too much mass to you. You're going to fall right through. You're breaking those hydrogen bonds. So this cohesion is the ability to resist hydrogen bonds under tension. So light molecules are sticking to light molecules. Water sticking to water. So water molecules sticking together. So once again, that bug walking across water is an example of cohesion. Adhesion is the ability to stick to other polar molecules. So the attraction to the unlike. First year bio, when you guys did microscopes and you had slides. If you had me, I always said, when you rinse your slides, lay them out flat. Do not stack them on top of one another. Because what happens is you have that glass slide, you have a little water on it, your partner comes along, they stack the slide right on top. Well, that's adhesion. You have those kind of unlike molecules sticking together now, and it's almost like glue between them. So the water's going to cling to it. We see it in plants. Plants get watered, water goes down to the roots. Well, that water is going to make its way up to the leaves because that's where photosynthesis occurs. And photosynthesis needs water to occur. So that water is going to use adhesion. It's going to cling to the veins within that plant cell, working its way up. So the water sticking to the veins, unlike molecules there, unlike structures, are going to go through and be adhesion. That's also an example of capillary action. And you guys saw this within your chromatography lab that we did last week, that formal lab write-up. Water molecules moving up against gravity. So when you took that little black dot and you guys put it on your paper and you put that paper in water, that water traveled up. That solvent traveled all the way up and hit that solute to make a solution. Then that solution traveled all the way up the paper against gravity. So we have a little adhesion, the water molecules are sticking to the paper and they're moving up against gravity there. Inhibition means drinking up. So if you garden by any chance and you plant your little seeds in the ground and you water the soil, that water gets sucked up into the seeds so that those seeds can germinate, meaning they start to burst and make little roots. That's inhibition. You also see it with a sponge that soaks up water. So you have water, you're wiping down countertops, it's soaking it up. Or when you get out of the shower and you have a towel and you're drying your hair, that towel is pulling out or sucking up that water. We also say because of these hydrogen bonds, water is resistant to temperature change. Now this is huge within our ecosystems and within our environments for living things. We say that water has a high specific heat. That means the amount of energy required to raise the temperature by 1 degree Celsius. It takes a lot of energy to make water raise by 1 degree Celsius. Well, that's great. Think about the days during the summer where it was like 100 degrees outside. If those lakes and streams and ponds and oceans, if we didn't have this high specific heat and they heated up very quickly, everything within them is going to die. It's going to get too hot for them. So all of our living organisms that live in that water would not be able to survive. So because of that, we're able to do that. So the specific heat of water, just a little fact for you guys, is actually 5 times greater than sand. So think about it. On a hot summer day, the beach sand may quickly warm up to a point where it's too hot to stand on. So I know I went to the beach for 1 week this summer, went down, get to the end of the little boardwalk or plank to get to the sand. You usually take off your flip-flops because it's impossible to walk in flip-flops in the sand, and your feet are burning because that sand is super hot to stand on. While the ocean water warms only a little bit. So imagine if the ocean heated up as quickly as that sand did. It'd be too hot to even go into. During the evening, the temperature of the sand decreases while the temperature of the ocean remains relatively constant. So because of that specific heat, it takes longer, it's not only to increase, but also to decrease. The same thing with the sand that we have there. So large bodies of water have relatively constant temperatures, constant environmental temperatures. It means it helps us to maintain our homeostasis. It's one of those words in first year bio that you guys should definitely know. Homeostasis, we'll see it again on the next page, basically means balance, internal balance, whether it's water temperature, body temperature, populations of organisms. We want to make sure things stay within balance. So our body temperature, 98.6, that's our homeostasis. I don't care if I pop you outside on a freezing cold day or I put you in the blistering heat, you should maintain homeostasis, internal balance for your body temperature. Water has a high melting point. Crystal pattern of ice is either 32 degrees Fahrenheit or 0 degrees Celsius. And we're going to talk about the difference or the importance of that on the next page as well. It also has a high boiling point. That's important because it takes so much to raise that temperature and to get there to boil that if it was a lot lower, it would evaporate and we would lose a lot more water. So because we have that high boiling point, it makes evaporation slow down just a little bit for us. Third page. So most energy from heat is used to break those hydrogen bonds instead of causing the molecules to move in order to raise the temperature. And that is why we have that high boiling point there. I just mentioned the word homeostasis, internal balance. It's kind of like that word in first week of school for biology, your first year biology that you went through. Things that you should know. Ability to maintain a steady state despite changing conditions. And that's where I said it. Pop you outside in the cold, it pop you outside in the heat, you're maintaining that body temperature. And water is important in this process. Think about it. It makes a good insulator. It resists temperature changes. It's a universal solvent. It can be used as a coolant. And think about this. Ice protects against temperature extremes. We just said that this ice crystal pattern forms at 32 degrees Fahrenheit or 0 degrees Celsius. But we also know that water is most dense at 4 degrees. So 4 degrees Celsius water is going to sink. 0 degrees Celsius water is going to be at the top, and that's where ice is forming. So ice on a lake or pond is going to form from the top and work its way down. If it went the other way and ice formed from the bottom and worked its way up, think about all the fish and sea critters that are now getting pushed up to the surface and that eventually are going to die. By having ice form at the top, 0 degrees Celsius, and my 4 degrees Celsius water down the bottom, those organisms can hover down towards the bottom. They're kind of insulated under that layer of ice there. As we go through and within your chromatography lab, you guys also saw things like water as a solvent. Water is a solvent. It makes up a large part of the solution. It is the liquid portion of it. A substance dissolved in water is the solute. So you might have one little typo there that you guys might have to fix. The solute is a lesser quality. So you're going through, you're making iced tea mix, you go home from school, you decide to make that iced tea mix, you got your water, that is your solvent, you put your iced tea mix in, that powder mix, that is your solute, you mix it all up together, there's your solution. Chromatography lab. You grab your piece of chromatography paper, you put your black dot right on it, you put it into water. Once again, water is still a solvent. That black magic marker that we have was our solute. What happened when they came together? They went into solution. Solution is what moved up that paper, adhesion, capillary action, in order to create those different colored bands for you that you guys should be measuring. We know that water dissolves a few things. It dissolves ionic compounds such as salt, sodium chloride, breaks it into ions. We have polar covalent compounds such as ethanol, and there's your formula for that, C2H5OH, and then small organic molecules such as sugar. So we said those are the ones that are kind of hydrophilic, water-loving. With this section, we also talk a little bit about acids and bases as well as buffers. So acids and bases are probably something you guys did in chemistry if you've had chemistry. But buffers are something that we talk more in human bio because it's what helps maintain our homeostasis within our body. So we know that hydrogen ions, water is H2O, so we have hydrogens there, and if we take that hydrogen away, we create ions. Hydrogen ions can affect cell structure because it affects the pH, whether something is acidic or basic or alkaline. So when water breaks apart, it dissociates, it breaks apart into hydrogen ions, which are H pluses, and hydroxide ions, which are OH negatives there. So how does that affect the body? Well, let's think about our pH scale first. You guys have seen this before. pH scale goes 0 to 14. We know smack dab in the middle is neutral. That's pure water. H2O, I have the same number of hydrogens as I do hydroxides. It's balanced, it's even. If I go from 7 to 6 to 5 to 4 to 3 to 2, it becomes more and more and more acidic. I'm gaining more and more hydrogen ions. So I know that 2 is going to have more hydrogen ions than a pH of 4. If I go the other way, I'm now looking at hydroxide ions. I gain more hydroxide ions as I move my way up. So my strongest base is going to have the most hydroxide ions than my acid will. And these alkalines or bases will go from anywhere from 7.1 all the way up to 14. So pH is actually the measure of concentration of hydrogen ions in solution. Do I have a whole bunch of hydrogen ions in solution, or do I have very few? If I have very few hydrogen ions, that means I have a ton of hydroxide ions. Here's some examples for you guys. We know that 0 to 1 being the most acidic, 13 to 14 being the most basic, 7 is neutral. So things like stomach acid. Yep, your stomach acid, which is able to break things down, is very acidic. It's got about a pH of 2. Remember, you guys have mucus that lines that stomach. That's going to protect those cells. Those people who get stomach ulcers, for some reason that mucus is not quite doing its job, and maybe that stomach acid's kind of burning its way through the stomach. That's what a stomach ulcer is. So when you take things like milk of magnesia, or Tums, they're very, very basic. So if I throw milk of magnesia or Tums into my stomach, which has a pH of 2, I'm basically trying to raise the pH so that it doesn't hurt my stomach as much. I'm trying to calm it down. I'm trying to neutralize it just a little. Vinegar is for your saliva, has about a pH of 8, so it's slightly basic, and bleach has a pH of about 13. A lot of the things within our body is going to be within this kind of like 6.4 to 8 range. So you guys eventually be doing a urine lab, and we'll be talking about the acidity, and you guys will be testing pH of different urine samples, synthetic urine of course. We'll talk a little bit more about the blood and the pH of that particular blood, as well as other things within the body. When we move on that pH scale, just so that you are aware, we go, oh, we have a pH of 6, and then we have a pH of 5. Oh, it's only 1 over on the scale. It's actually a lot more than 1 over on the scale. So each unit corresponds to a 10-fold increase or decrease of hydrogen ions. So here's my pH of 7. Okay, so if I go from a pH of 7 to 6, pH of 6 has 10 times more hydrogen ions than 7. If I go from 7 to 5, I'm now at 100 times more hydrogen ions. 7 to 4, I'm at 1,000 times. I'm at 10,000, and it keeps going. The same happens when I go more towards my alkaline side, except now I'm going 10-fold of hydroxide ions. 7 to 8, 10 times more hydroxide ions. 7 to 9, 10 times more. So it's not just 1 over and you don't have to worry about it, it's not that bad. It's a 10-fold increase every time we move through there. With these acids and bases, some things that you guys should know is that acids are all substances that donate hydrogen ions. What we just said, the more you go acidic, the more hydrogen ions you have in solution. Bases are ones that are going to basically give off hydroxide ions there, or that are going to accept those hydrogen ions. So a solution with a greater concentration of hydrogen ions is acidic. A solution with a greater concentration of hydroxide ions is alkaline or basic. There are a few ways that you should be able to test these. First one is a pH meter. You go to the water. If you are an environmental scientist, you have this little, almost looks like a little calculator. It's got a little probe. You stick it in the water. It gives you a number of 8.2. And it might be even a little more exact than that. This provides quantitative data. It's going to give you numbers. It's super specific. Maybe you have litmus paper, and litmus paper is a little roll of paper and it's orange, and you basically pull it over. Let me grab a roll for you real quick. So it's a little roll. It looks like this. You're going to pull out a little piece of paper. You dip it into your substance. It's going to change color on you. Once it changes color, you guys will then be matching it up to the color wheel that you have on here. This one's a little bit more specific. I have ones that go from 1 to 14. This one's more specific for when you get to urine. This is more our body range, so I want to be a little more specific in numbers. This goes 5.5 to 8, and I would just kind of go through and match my color once I put it in there. So you guys will see both types, the 0 to 14 litmus paper, as well as our pea hydrogen paper. So I'm going to backtrack. That was pea hydrogen paper that you guys just had there. Litmus paper does not really tell you guys much. Litmus paper, you guys probably did in first year bio. You have red litmus paper, you have blue litmus paper. You're going to take both, you're going to dunk it into something. If red stays red and blue turns red, you are dealing with an acid. If you take red and blue and you dunk it into an unknown substance and blue stays blue and red turns blue, you're dealing with a base. So anything that's blue is usually going to be a base. Anything that's red is usually going to be an acid. This is qualitative. It's not going to give you a number, that litmus paper. These colors will match up with your pea hydrogen paper that we have here too. So anything, usually your reds and your pinks are acids on that particular color code. Anything that's more bluish and green is going to be your bases there. So pea hydrogen paper, the wheel that we have here that you'd be matching up, is both quantitative as well as qualitative within that. Buffers. Buffers are going to keep the pH in balance by regulating the level of free hydrogen ions and hydroxide ions within your body. So we said a lot of your body fluids like to be right around that neutral range. Stomach acid is a little different. Body fluid needs to stay in balance. So for instance, cytoplasm that makes up your cells is about 7.2. Well, within your cells, you guys have these organelles called lysosomes. They're basically like the stomachs of the cell. They break down old cell parts for your bacteria. Their pH is a 5. They have digestive enzymes inside of it, kind of like your stomach. So organelles have their own pH, and we need to maintain these pHs, even your blood, anywhere from about 6.8 to 7.2. So your body has this way to maintain it. It uses these things called buffers to help minimize pH change. So any dramatic shift in pH can play a role in controlling cellular activities. So for instance, if we start changing pH on things, it can affect things such as cell division after fertilization. It can lead to certain problems with that embryo starting to form. Cells are going to maintain this pH within the body, that normal within range, by using buffers. If this doesn't happen, let's think about the blood, and we said the blood likes to stay close to neutral there, anywhere slightly above or below is okay, not too much. But if it becomes too acidic, you have too many hydrogen ions within solution, what happens is it goes through acidosis. And this happens when you have too many ketones within the body. So ketones are kind of acidic, and we see this a lot with diabetics. Their body's not able to regulate it. They're not able to make that insulin to take care of those ketones, and they can get their blood become too many hydrogen ions. It becomes too acidic, and they can lead to acidosis, which is why they have to monitor their blood sugars so well. If we go the other way, alkalosis is too basic. This can lead to convulsions, it can lead to excitability of the nervous system, it can lead to muscle spasms. So there's things like if you think about respiratory alkalosis, so I'm going to go down here a little bit. That means we have low carbon dioxide levels within the blood. We have low carbon dioxide levels in the blood because maybe you have a lack of oxygen, maybe you have some kind of lung disease, maybe some kind of medications are poisoning within your body, some sort of liver disease. And what that does is it leads to this basic pH within your system. It lowers everything, and you basically start to go into convulsions. You can have problems with your nervous system. You start to have muscle spasms. If untreated, it can lead to death. So weak acids or bases that react to strong acids or bases prevent sudden changes. This is called a neutralization reaction. Go back to the Tums scenario I said with stomach ulcers. Your stomach is a pH of 2. It's a strong acid. I'm going to throw in a base. So if I mix a base with an acid, I'm helping to bring it closer to 7. I might not bring it exactly to 7, so neutralization doesn't mean that it has to be a pH of 7. It just means I'm bringing it closer to neutral so that the body is able to handle it. So these buffers, these weak acids and bases, are produced naturally in the body to maintain this homeostasis, this balance, this pH that it should be within the body. So what we see here usually is that when a strong acid and base interact, a salt is going to be formed, and this is what we do with neutralization. One of our primary buffers within the body is bicarbonate salt. It works by donating protons in the blood if it's too basic, and accepting protons if the substances are too acidic. So think about it, a little FYI for you. When you're exercising, your blood becomes more acidic. Carbon dioxide, you're breathing heavier. The protons are absorbed by the bicarbonate in your blood to form carbonic acid. This causes your lungs to expel more CO2. You're breathing heavier, you're releasing more CO2, which causes the acid in your blood to be lowered back to normal. If you don't exhale that CO2 out of your system, your blood's going to become more acidic, carbonic acid within your body. So that bicarbonate salt is helping to kind of make sure that that pH stays normal. So as you guys went through this, you should have highlighted any questions that you have. You guys will be looking at some stuff with watercolor as well as water questions. This due dates on those things I'll make a little bit later after I come back. That way if you do have questions, you guys can ask me. You guys can email me, and we can probably FaceTime a little bit later in the week. We can FaceTime just to make sure that if you guys do have questions, we can get them covered. Okay, so hopefully this helps you guys out a little bit, and I hope to see you soon.